patients with sickle cell disease have a single missense mutation in the hemoglobin beta gene, HBB, which encodes the protein beta globin. This mutation results in production of sickle hemoglobin, HbS, that polymerizes when deoxygenated, which hinders blood flow, damages myriad tissues and organs, causes severe pain, and also shortens the survival of red blood cells, resulting in anemia. Patients with transfusion-dependent beta thalassemias have mutations in HBB or in its regulatory regions that lead to impaired or absent production of beta globin in developing red blood cells, ineffective erythropoiesis and, thus, transfusion-dependent anemia. Great strides have been made in allogeneic hematopoietic stem and progenitor cell, HSPC, transplantation for both disorders, but the majority of patients with sickle cell disease or transfusion-dependent beta thalassemias lack suitable family donors who are unaffected. Following decades of setbacks since the first development of viral gene transfer vectors, HSPC gene therapies that use integrating lentiviral vectors have recently made remarkable strides in the treatment of a number of human blood and immune disorders, including the successful delivery of anti-sickling or replacement globin proteins to HSPCs for the treatment of sickle cell disease and transfusion-dependent beta thalassemias. The application of genetic therapies to hemoglobinopathies has faced much higher hurdles than has the application of such therapies to immunodeficiency disorders, for which modification of a small fraction of multipotent HSPCs or committed lymphoid progenitors can result in sustained improvement, due to the in vivo selection of corrected versus mutant cells, and/or the ability of even a minor fraction of corrected cells to reverse immunodeficiency disorders. First, to prevent sickling in sickle cell disease or to normalize red blood cell production in transfusion-dependent beta thalassemias, individual erythroid cells must contain very high concentrations of anti-sickling globin proteins, such as fetal hemoglobin, HbF, or sufficient replacement globin chains in the case of transfusion-dependent beta thalassemias. In addition, at least 20% of HSPCs must reach this threshold to reverse the disease phenotypes, given the results from patients assessed after allogeneic transplantation. Second, expression of the therapeutic globin must be lineage-specific. Third, the genotoxic consequences of integration of the viral vector into the genome and the serious toxicities of conditioning regimens given before transplantation of genetically modified cells are more concerning problems in diseases such as sickle cell disease and transfusion-dependent beta thalassemias that are not immediately fatal, and must be mitigated before widespread utilization. There has been a flurry of promising results in several recent clinical trials using lentiviral vectors for gene delivery. These results are thought to be due to the specific properties of these vectors relative to those of earlier approaches. High, erythroid-specific expression of beta globin after highly efficient lentiviral transduction of HSPCs results in transfusion independence in the majority of patients with transfusion-dependent beta thalassemias. In addition, the reversal of biological markers of sickle cell disease and a lack of subsequent vaso-occlusive crises were reported in a single patient with sickle cell disease after lentiviral introduction of an engineered anti-sickling beta globin into the patient's HSPCs. A larger trial of this approach is ongoing, with very encouraging data reported at meetings. Modern lentiviral vectors have proven less genotoxic than the mouse retroviral vectors that were previously used in immunodeficiency gene therapy trials and were linked to genotoxic leukemias. However, the first patient with transfusion-dependent beta thalassemias who received lentiviral gene therapy developed a concerning but thus far benign clonal expansion linked to vector-induced aberrant splicing of a gene encoding a protein involved in growth control. This has provided further impetus for the development of targeted gene editing approaches to avoid semi-random vector integration events. The next generation of gene therapies has taken advantage of the observation that hereditary persistence of HbF ameliorates the clinical manifestations of both sickle cell disease and transfusion-dependent beta thalassemias. The transcription factor BCL11A is known to turn off fetal gamma globin and turn on expression of the gene encoding adult beta globin postnatally. Turning off BCL11A in erythroid cells was shown to result in high expression of HbF and decreased expression of adult hemoglobins, including HbS. In a recent trial of 6 patients with sickle cell disease, a lentiviral vector expressing a short hairpin RNA targeting BCL11A, embedded in a microRNA, SHME-R, was expressed from an erythroid-specific promoter and was introduced into HSPCs to turn off BCL11A specifically in developing erythroid cells. This resulted in the expression of HbF in a large percentage of red blood cells, F cells, as well as a high concentration of HbF per cell after transplantation, with marked clinical improvements in the majority of patients. Now the scientists describe the first clinical use of CRISPR-Cas9 to edit HSPCs in humans, specifically to treat hemoglobinopathies. They used this new technology to aim for the same downstream effects obtained in the trial of lentiviral SHME-R described above. Erythroid-specific decrease in BCL11A expression and, thus, reactivation of HbF. The authors published results obtained for one patient with sickle cell disease and one with transfusion-dependent beta thalassemias enrolled in an ongoing larger trial. They use electroporation to introduce Cas9 protein and a guide RNA targeting the BCL11A erythroid-specific enhancer into HSPCs from these patients. The authors' aim was to induce non-homologous end joining repair, in order to create small insertions and deletions, indels, precisely at the binding site, located in this enhancer, for the transcription factor GATA1. This prevents enhancer function and, thus, expression of BCL11A in developing erythroid cells, which are normally dependent on GATA1-induced BCL11A expression to shut off HbF. The patients underwent myeloablation via busulfan, followed by infusion of edited HSPCs, and in both of the patients, the edited HSPCs engrafted, albeit somewhat more slowly than expected, suggestive of some loss of HSPCs due to editing. The patients reported in the paper had 69 to 85% indels at the target site, which resulted in considerable production of HbF, transfusion independence, resolution of anemia and no further vaso-occlusive crises in the patient with sickle cell disease. Notably, the patients are part of a larger trial of this approach, with similarly encouraging results reported now in a total of 9 patients with hemoglobinopathies. Targeted gene editing is expected to be much more specific and less likely to result in genotoxicity than as semi-random integration of lentiviral vectors. However, off-target editing can occur due to homology of the guide RNA sequence with additional genomic sites. The investigators made a comprehensive effort to identify susceptible off-target sites through the use of both an algorithm based on sequence similarities and an unbiased method that predicts sites via retrieval of nuclease-induced double-stranded DNA breaks from live cells. Deep sequencing of all predicted off-target sites was performed on edited control CD34 plus HSPCs prior to clinical trial initiation, and no off-target editing at any site was detected. However, a similar analysis of HSPCs or blood cells from the clinical trial has not been published, and this will be of critical importance, given the potential for even very rare off-target events to result in changes in HSPC behavior. In conclusion, there are now at least 3 promising gene therapy approaches likely able to cure sickle cell disease and transfusion-dependent beta thalassemias. While CRISPR-Cas editing theoretically is a more targeted and thus safer and more elegant approach, much longer follow-up for both efficacy and safety must occur for a valid comparison with the more mature results from lentiviral gene addition or SHME-R BCL11A knockdown approaches. Additionally, methods now exist for actual correction of the point mutation associated with sickle cell disease. CRISPR-Cas-induced homology-directed repair of this mutation can theoretically occur via co-delivery of a corrective homologous cassette. However, issues remain to be solved, including low-efficiency homology-directed repair that results in mutation correction, in contrast to more efficient indel production via non-homologous end-joining repair, and depletion of self-renewing HSPCs due to activation of the transcription factor p53 that results from double-stranded DNA breaks. Another approach known as base editing, guide RNA targeted localization of an enzymatic complex able to correct or alter specific base pairs, is even more attractive, due to the lack of any occurrence of double-stranded DNA breaks via this editing method, and it has been already used to inactivate the BCL11A erythroid enhancer in preclinical studies. Design of a base editor able to directly correct the mutation associated with sickle cell disease is being pursued. The future is bright but also complex, both in terms of comparing and optimizing all of these approaches and in delivering them to patients in a cost-effective and safe way.